Good afternoon, everyone. Welcome to TeleShadowing. We are live on YouTube. Feel free to ask questions in the chat box as we go along, and your questions will be addressed. So today I'm very excited to kick off our Pathway to Med Ed or Medical Education series, where we're basically going to be talking about the pre-clerkship curriculum, kind of learning from a bird's eye view in a clinical sense and understand the clinical relevance behind a lot of different diseases and things that you learn in your pre-medical courses that are required for medical school. So I hope this will be beneficial to you as you're preparing for medical school so that you can be white coat ready. So again, we're going to start off with our biochemistry and clinical applications month for the month of August 2024, and it will be continuing on all the way till, I believe, February or March 2025. I'm going through all the major topics of the USMLE First Aid Step 1 book. So, all right, let's kick it off with talking about clinical applications of the central dogma. Now, just to remind you all, the central dogma is basically the fundamental basis of life, and I'll get into detail as to what I mean by that. So today we'll learn about biochemistry basics, learn just to revise again and revisit what is DNA, what is the purpose of DNA, what is the genetic code and its features. We'll learn about DNA more specifically, the transcription, translation, DNA repair, some different mutations that can happen when you do DNA replication. And then we'll learn about the central dogma itself, from transcription to translation to protein, and what its clinical applications and why it's important. So let's actually learn more about biochemistry basics. So again, this is one of the most fundamental principles in biology, the central dogma. It's because it's not just I guess you could say, a cornerstone of molecular biology, but it's also the basis of understanding how genetic information is transferred within a cell to produce the proteins that are very essential for life. Remember, amino acids are very high yield on the MCAT. Amino acids are the building blocks of protein. So I hope that makes a little bit more sense as we kind of see it, and in zooming out to big picture, like, why do I care about this? Well, without RNA giving you the instructions for the different codons that we'll get to, you won't know which amino acids should make what types of proteins for the different functionalities of the body. So the central dogma of molecular biology basically describes the flow of genetic information within the biological system, and it's typically summarized as going from DNA to RNA to protein. So the DNA is in your nucleus of the cells, then the RNA, the mRNA actually gets transcribed in the nucleus. Transcribed means kind of copy pasted, and then the pre-mRNA gets processed, and the mRNA actually leaves the nucleus, as it's called, messenger RNA. And once it leaves the nucleus into the cytosol, that's where it kind of attaches itself to a ribosome with the help of other types of RNA like tRNA, rRNA, etc. So we'll learn a little bit more about that in much more depth, but that's just sort of giving you the general overview of what we're going to be discussing. So it's basically this small, it's a simple statement, right? DNA to RNA to protein, and basically encapsulates the process by which the genetic code that we have that's stored in our DNA is essentially used to create the proteins that carry out nearly all of the functions that are within our cells. So how it works is the DNA is basically the material that we inherit from our parents, and almost all other organisms also have DNA. It has the instructions that are necessary for us to develop, to survive, and reproduce. And the instructions, or the instruction manual, is found within the DNA sequence of nucleotides. If you'll remember, the nucleotides, there are two types of nucleotides. There's purines and then there's pyrimidines. Purines include I'm just trying to remember, cut the ring. That's basically the acronym, the mnemonic used to remember. So cut stands for cytosine, uracil, and thymine. I believe those are the pyrimidines. And then the purines are adenine and guanine. So then there is transcription. Transcription is DNA to RNA. DNA to RNA is the first step in the central dogma. And it's basically the first step of getting the DNA to where it wants to go. So let me actually, yeah, okay. So this process of transcription is carried out by RNA polymerase, and that reads the DNA strand, and then that synthesizes a complementary strand of RNA. And I'll show you a couple of diagrams about that. And the RNA that comes out of that is the mRNA or messenger RNA, and that's like a temporary copy of the genetic information that's needed for protein synthesis. And maybe you'll remember this from maybe high school biology or maybe your molecular biology classes in freshman or sophomore year of undergrad, where basically remember that RNA is different from DNA in one major thing. Of course, DNA is double-stranded, RNA is single-stranded, but also another difference is that RNA has uracil, whereas DNA has thymine. So that's sort of another difference. Instead of thymine in DNA, the RNA would have uracil. They're very similar in composition. So that's why we have that additional type um, of a pyrimidine. So now when you want to go from RNA to protein, the mRNA is actually then used as a template to build a protein during the process of translation, and that occurs in the ribosome, in the cytosol or cytoplasm, and that's basically where the mRNA is decoded by transfer RNA or tRNA molecules, and then they bring the appropriate amino acids to build the protein. And so the sequence of nucleotides in the mRNA actually determines the sequence of the amino acids in the protein, and therefore its structure and function. Because remember, the amino acids, they determine what type of protein it is, whether it's primary, secondary, tertiary, quaternary in structure. So again, this is all a review of undergrad biology. That's why I'm just able to just kind of spitball from memory, because this is all prerequisite information to know before we start medical school. So why do we care about this? Why is this important? So the central dogma is very important for many different reasons, but primarily because it helps to understand genetic disorders. Like if there are any mutations or errors in the DNA sequence, they can actually lead to changes in the mRNA, and therefore the protein that gets produced from that mRNA, right? So the changes that can disrupt the protein's function would lead to genetic disorders. For example, if there's a single point mutation, which we'll learn about more in depth in a couple of slides, there's a single point mutation in the gene for hemoglobin that will lead to sickle cell anemia. And that's a condition where the altered hemoglobin protein causes the red blood cells to adopt to a more sickle shape that you see in blood smears under a microscope. And that leads to various health issues. So again, there's this I remember feeling very disconnected when I didn't understand why I was learning all of this transcription, translation, and in a lot of detail for the MCAT and also for my undergrad classes. It's because it's very relevant to clinical medicine, how we practice it, and how diseases manifest in patients. So molecular biology is almost maybe even more important, just as important as the macro anatomy and things like that. So another reason why the central dogma is very important is because a lot of diseases derive from issues that happen, because when you know what's normal, then you know what's abnormal. So like diseases like cancer, they're driven by mutations in DNA that actually cause the abnormal proteins to be produced. So understanding how these mutations affect the flow of information from DNA to protein actually helps us understand better the mechanisms of disease. And I would say a third reason for why it's super important is like targeted therapies and also biotechnology applications. I would say it's very important for those things because it provides this framework for developing targeted therapies. Like if there's a disease that's caused by a specific protein that's malfunctioned, then scientists can design drugs to target that protein or prevent that protein from even being developed. Or basically from being before it's even formed, or try to denature that protein, but you want to know where it's being made or what genes are responsible for it. So this approach is the basis of many different modern therapies, including those for cancer. So like there's a drug called Herceptin that specifically targets the HER2 or HER2 protein that's overexpressed in some types of breast cancers. And there's also like biotechnology applications, such as when there's insulin used in diabetes treatment, is actually produced by inserting the human insulin gene into a bacteria. That's a process that's based on the principles of the central dogma using like CRISPR-Cas9 techniques. And that actually allows bacteria to produce human insulin that can be harvested and then used as medication. So I would say there are so many applications for this serving as more than just a biological concept, is also like a foundation of how life operates at the molecular level. So when you understand this flow of genetic information from DNA to RNA to protein, you can actually learn some very important insights into like how genetic disorders might happen. And we're actually going to be learning more about that in detail next week, like hereditary diseases, how a disease can actually progress, and also how you can develop more effective and targeted treatments. So it's not only enhancing our understanding of biology, but also has very practical implications for medicine and clinical medicine and patient care and biotechnology. And so that's why it's very important for anyone who wants to pursue a career in medicine, because you're going to be able to see that in patients, and also in really any type of research you do. A lot of research is based on this as well. So we're going to explore some different ways the central dogma applies, right? So we're going to learn about replication, we're going to learn about transcription and translation, and how a gene becomes a protein and why it's very important. So again, this is just a review, right, from what we should already know. Or maybe you're just learning this for the first time, and that's completely okay if you're still in high school or earlier on in your pre-med career. It never hurts to apply something called spiral learning that we learned about last week in medical pedagogy. So basically, if I mention something once, I will revisit it again, and that's called spiral learning. And every time I talk about it, I'll increase in the depth I talk about it. So again, just an overview of the genetic code. So the genetic code is basically, it has a set of rules, like any instruction manual would, by which the information that's encoded within genetic material, like DNA or mRNA sequences, is translated into proteins by living cells. So let's actually like talk about the features of the genetic code. So we'll focus on its universality, the redundancy, also known as degeneracy or being degenerate, and the roles of start and stop codons in protein synthesis. So let's start off with the point on the left, universality and redundancy. So what actually comes to mind when we hear these words? So when I hear of universality, I think of being able to be applied to many different contexts, and being universal across all organisms, which is true because the genetic code is nearly universal across all organisms, from the simplest bacteria to very complex multicellular organisms like us, humans. So this universality actually indicates that all life on Earth um, uh, is actually very applicable to genetic engineering, and it helps us make the use of model organisms and research very possible. And then redundancy or degeneracy, the genetic code is actually described as degenerate because most amino acids are encoded by more than one codon. And I'll show you, I think here it is. Yeah, this is the codon dictionary, if you will. So for example, the amino acid leucine, it's encoded by 6 different codons. And so this redundancy is crucial for the resilience of the genetic code against mutations. So because there are all these codons that can, for example, code for arginine, if there is, for example, and by accident, there's a uracil encoded instead of a cytosine, it'll still encode for the same amino acid. And that's very helpful because your DNA is replicating at billions of times per minute. Remember, DNA is just one part of your billions and trillions of cells. So, and if you're just wiping your hand, and that's actually just like deleting more and more cells and more dead cells and more dead skin cells, and cells make up every single part of your body. Remember, cells make up tissues, tissues make up organs, and organs make up organ systems, and organ systems make up organisms. So cells are the basic foundation in the nucleus, and inside the nucleus includes the DNA. So again, this is happening trillions and trillions of times. And so it's really important to have this redundancy, because that really helps with the resilience of the genetic code against mutations. And this is a sidebar, but if anyone has ever done like a 23andMe or done like genetic testing before, you can actually download your raw data and you can actually read your specific codons in your DNA, which is very interesting. But let's get back to the significance of redundancy. It's really to help protect against mutations. So, if a mutation occurs usually in the third base of a codon while it's being translated, and I'll show you that process. There's a lot of really cool videos about it that I'm excited to show you all. it actually might not change the amino acid that's being incorporated into the protein. Therefore, it reduces the potential harmful effects of point mutations. So again, like if there's GAA, which encodes for glutamic acid, and it mutates to, let's say, GAG. So there, instead of a GAA, so instead of an adenosine, it goes into another guanine, GAG. It'll still code for the same amino acid, glutamic acid. So when you're looking at it from the big picture lens, when you're zooming out, there's no difference. So there's no difference in the protein. There'd be no change in that protein. And then you want another important aspect of the genetic code is the codon specificity and the start and stop codons. So basically, the start codon is typically AUG, which codes for the amino acid methionine. And that codon actually serves as the start signal for translation, which marks the site at which the ribosome begins making or synthesizing the protein. And this is important because it sets the reading frame for the ribosome, which ensures that the mRNA is read correctly, which it means, and by read correctly, I mean 3 nucleotides at a time, because a codon is made of 3 nucleotides, to produce the correct protein. And then the stop codons include UAA, UAG, and UGA. And these stop codons don't actually code for any amino acid, and instead they signal the termination of translation. So and that's how you can actually tell differences in protein sizes, because not all proteins are in the same shape. I mentioned before, there's primary, secondary, tertiary, and quaternary proteins. And so these codons are very important for defining like the proper end of that protein. And so the protein can be the correct length and has the proper sequence of amino acids. And this is super clinically relevant because if there are mutations that affect start codons, that can actually prevent a protein from being synthesized altogether, and that can lead to significant cellular dysfunction. And if there are mutations that generate like premature stop codons, that can result in a truncated or non-functional. Basically, a truncated protein means like it was cut off prematurely, which means it's a shortened protein from what it originally should have been. And that becomes like essentially a non-functional or dysfunctional protein. And that's very common. It's a very common cause of genetic disorders like Duchenne's muscular dystrophy. So we'll learn about that a little more. But again, I'm just throwing out these disease names. You don't have to learn those in depth. But again, you also need to understand the pathophysiology behind diseases in order to understand the treatment, in order to understand the treatment regimen and how patients can live better lives. And you really need to understand the basic reason for why something happens. Having that understanding will help us understand what is the pathophysiology of disease, like Duchenne's muscular dystrophy. So now let's talk about DNA, specifically DNA replication. So DNA replication is basically a process that occurs every time a cell divides. That billions of trillions of times that's already occurred, even more than trillions. That basically ensures that each daughter cell receives an identical copy of the DNA. Because, for example, your palm cells are very different from the cells, your cardiomyocytes or your heart cells. Now, I don't want to be growing heart cells where my palm of my hand should be. So that's why I need identical copies of DNA. And again, form follows function. So wherever the DNA is surrounded by or wherever it's supposed to be, I want palm, hand cells. I want skin cells. I want epithelial cells. I don't want heart cells growing in places they're not supposed to be growing. And one really interesting fact I learned when I was in high school biology is that every cell has the DNA that you need. So for example, because they all derive from the same stem cell, because remember, we're all embryos once. So technically every single cell has the potential to be a heart cell or a skin cell, but there are specific genes that are muted, I guess you could say. And I'll explain what muted means in a couple of slides. So just something to think about. Like, so basically every cell in my body has the potential to grow into a heart, even if it is from my eyeball. But why is it that my eyeball cells stay like an eyeball, and why is it that my heart stays like a functioning like made up of cardiomyocytes? And we'll learn about that. It's just something very interesting when I learned why that is, because every cell has the genetic information it needs to be potentially those cells. But anyway, let's get back to DNA replication. So basically, DNA replication operates through something called a semi-conservative replication process. So basically, DNA replicates in a semi-conservative nature. What that means is that basically DNA is double-stranded, right? So DNA essentially gets zipped apart because it's, of course, in that spiral shape. When it gets zipped apart by a helicase, there are two daughter DNA strands. And so they consist of the original template strand and one newly synthesized strand. And so this method of replication actually conserves half of the original DNA molecule in each DNA molecule. And this is very important because it ensures that there's genetic consistency across all cellular divisions, and that maintains the integrity of the genetic information that's passed from one generation of cells to the next. So there's DNA polymerase, and that's responsible for, that's an enzyme. Anything that ends in A's or ligase, helicase, polymerase, those are enzymes. And enzymes are basically responsible for like cutting things, chopping things, or making things work, or basically enzymes help reactions go faster. That's, I guess, the layman way of explaining enzymes now that I'm not taking chemistry classes anymore. But basically, the DNA polymerase adds nucleotides one by one into the growing DNA strand that's complementary to the template strand. And that works, if you remember, the 5' to 3' direction, meaning it adds nucleotides to the 3' end of the growing strand. And DNA polymerase also proofreads. So it has a proofreading ability to basically double check. It's basically like your Grammarly of DNA replication. It basically allows it to remove any incorrectly, it can remove, or also the technical term is called excise, remove or excise, EXCISE, incorrectly paired nucleotides, and it replaces them with the correct ones, and that helps them to maintain high fidelity or high conservation during the replication. Then there's an enzyme called helicase. Helicase is one of my favorite enzymes because I think of it as like a zipper. So basically, helicase is the enzyme that unwinds the double-stranded DNA, which creates the two single strands that can be used as templates for replication. And this unwinding forms the replication fork, where the process of DNA synthesis occurs. And so when you separate the two strands, helicase is essentially assuring that the replication machinery has access, like the DNA polymerase, for example, has access to the template strands that are necessary for synthesizing the new DNA. And then there is our friend ligase, and ligase, I think of it as the molecular glue, and that's because DNA ligase is responsible for sealing the nicks between, if you remember, the Okazaki fragments in the lagging strand. So remember, there's a leading strand and a lagging strand. The leading strand doesn't have any breaks in it; it just zips through, because there's difference in the 5' to 3' difference. But then there's a lagging strand, and each like fragment is called Okazaki fragment. That's spelled Okazaki, Okazaki fragment, due to the 5' to 3' synthesis directionality. And ligase is very important for linking these Okazaki fragments together, which ensures the integrity and the continuity of the newly synthesized strand once the DNA has been replicated into those two new daughter strands. But remember, there is semi-conservation, which means that the original strand is still retained half-half in each new strand. So this is clinically relevant because of its role in cancer and also chemotherapy. Because if, for example, if there are any like errors in replication, if they're not corrected, that can lead to mutations. And so if there are more and more mutations that accumulate, it can actually disrupt the normal function of the genes, and that can potentially lead to uncontrolled cell growth, which is one of the hallmarks of cancer that we've learned already during our pathology month. So, like, for example, there are mutations in genes that control cell division, like tumor suppressor genes, and maybe P53 is ringing a bell. That can actually lead to the development of cancer if these errors are not corrected during replication. There are also chemotherapy drugs that target DNA replication, and they're basically designed to target the rapidly dividing cancer cells by interfering with DNA replication. So that's why understanding it is very important, because there are many ways to combat it or stop it from happening if it's happening abnormally or too much. So, for example, methotrexate is a therapeutic drug that's used to inhibit an enzyme called dihydrofolate reductase. And I'll show you what that looks like. And that's basically that enzyme is involved in the synthesis of nucleotides. And that way it would slow down the DNA replication, rapidly dividing cells. Because remember, DNA is made out of nucleotides, the purines and the pyrimidines. There's another drug called 5-fluorouracil that we'll revisit again, and that also acts as an inhibitor of another enzyme called thiamidylate synthase, and that will prevent the synthesis of thymidine, and that's also required for DNA synthesis, which also slows down or stops the replication process, because if there's no nucleotides, how are we going to replicate the DNA? So by targeting the DNA replication, these drugs basically aim to selectively kill cancer cells, which divide more frequently than the normal cells. So again, these drugs are able to target cancer cells, tumor cells, not normal functioning cells, because they just divide much more rapidly. Sometimes it can lead to side effects, and it can lead to affecting normally dividing cells, because again, DNA replication is happening in both types of cells. So the next step after understanding is finding therapeutics and doing the research into figuring out which ones will target the bad cells and not the good cells. And so that is why this is really what we're working on today, like how can we reduce side effects in normal cells. So again, here's a video that talks about it a little bit more on DNA replication. Let me reshare my screen with audio really quick. Got to do that. Here, share for sound, optimize video clip. Now you should be able to hear it. It's a 3-minute video just summarizing everything I just talked about.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay, I hope that sort of summarized everything we learned about just now, in a more succinct way. So again, remember the mention of semi-conservative, basically conserving half of the DNA on both ends. It's very important. Basically, these are just different, I guess you could say, guardrails to preventing mutation. Because remember, this is a process that does work. Not everyone has mutations, but in the case of mutations, it usually leads to cancer or uncontrolled growth, or mutations leading to other types of diseases like Duchenne's. But overall, in a healthy individual, it is working, and most of us, thankfully, are healthy. But in the case of the DNA not, working proper, DNA replication not happening appropriately, there are some mechanisms built. So this is another guardrail, not just the DNA polymerase double checking itself, but also there are DNA repair mechanisms. So these also maintain the integrity of the genome or the genetic code, and it ensures that the genetic information is accurately transmitted from one cell's generation to the next generation, because our DNA every day is actually exposed to many different damaging agents or carcinogens, like including there's UV radiation if you're going outside in the sun, there's chemical exposures, and even like the normal metabolic processes within the cell. So without efficient repair mechanisms, these damages could actually lead to mutations that may disrupt more gene function, and it could potentially lead to diseases like cancer. So let's actually explore the three different repair mechanisms. There's like single-stranded breaks and four double-stranded breaks, and then like the consequences of like what happens if these pathways are not functional or operational. So the first one is mismatch repair. The mismatch repair system basically corrects errors that escape the proofreading activity of DNA polymerase during the replication. And they typically involve like mismatches where incorrect nucleotides are incorporated into the newly synthesized DNA strand. And basically, mismatch repair proteins, they recognize the mismatch, they remove the incorrect nucleotide, and then they fill in the gap with a correct nucleotide using the complementary strand as a template. So you can see here on the next slide, on the in the middle, I believe that's what it's showing. Yeah, but if there's a base mismatch, that's basically what I described here. Um, there, for example, if it's two like pyrimidines paired together, or two purines paired together, that's a big no-no. You want to make sure that you're pairing purines with pyrimidines. So the next type is, well, actually, if mismatch repair is defective, then in the MMR system, and if there's any defects in this type of repair system, is actually associated with Lynch syndrome, also known as HNPCC. HNPCC stands for hereditary non-polyposis colorectal cancer. So patients with Lynch syndrome, they have a significantly increased risk of developing colorectal cancer and other types of cancer as well. And the defective repair mechanism allows for basically the accumulation of mutations, especially in the genes that are regulating cell growth and apoptosis, and that can actually lead to uncontrolled cell proliferation. Now, the second type of DNA repair mechanism I wanted to talk to you all about is the base excision repair, or BER. Now, base excision repair is basically responsible for fixing the non-helix distorting base lesions, like uh, usually the ones that are caused by oxidative stress or deamination or alkylation. And we'll learn about what is alkylation later on, but deamination and alkylation, remember, this is why organic chemistry is very relevant sometimes, or chemistry in general. I remember when I went through my undergrad courses, I had no clinical understanding why I was learning all of it, and so it was much harder to retain that information. I feel like I would have done much better if I knew, the clinical relevance for why I was doing these different things in chemistry and whatnot. But again, basic tissue repair has basically applications if there's a DNA molecule that's affected by a distorting base lesion, like I said before, like deamination, alkylation, and also oxidative stress. So if the lesions aren't corrected, it can result in point mutations, and we'll talk about point mutations in like 2 slides. But basically how base excision repair, or BER, works is the base that's damaged, it's essentially like recognized and removed by by an enzyme, and that enzyme is called glycosylase, and that creates an A basic site, and that A basic site is then removed or excised, and that gap is then filled and sealed by DNA polymerase and ligase. So remember our trusty, our handy friends, DNA polymerase and ligase being that molecular glue, and DNA polymerase being the one that actually, builds the building or puts the bricks together in the building. And how this is relevant clinically, if there are basic fusion repair, like if it's defective, if there are impairments in the BER system, it's linked to different conditions. Now, the first aid book didn't actually mention specific conditions, but what I could find online is like an increased susceptibility to neurodegenerative diseases such as Alzheimer's disease. So if there is this accumulation of unpaired of unrepaired oxidative DNA damage in neurons, that's actually thought to contribute to the pathogenesis of later neurodegenerative conditions like Alzheimer's. Now, the third type of DNA repair mechanism is called a nucleotide excision repair, or NER. And basically, it's responsible for repairing the bulky or helix distorting lesions that are caused by UV light primarily, or chemical exposure. Now, if the UV light causes a lesion like a thymine dimer, that's bad because, like I said before, that's two pyrimidines together. And these lesions can actually block transcription and replication, and that can pose a very serious threat to the cell's viability or ability to survive. So the NER system, it basically identifies any distortions in the DNA helix, and it removes a short single-stranded segment, like an entire segment as a whole, that contains that lesion and fills in that gap using a complementary strand as a template. And this is actually very clinically relevant. I see questions on this all the time when I'm doing like UWorld questions, like studying for a step. Basically, if there are mutations in the genes that are involved in NER, it can lead to disorders like xeroderma pigmentosum, XP. And it's because individuals who have XP actually have a heightened sensitivity to UV radiation, and that also leads to a dramatically increased risk of skin cancer. So without like a functional nucleotide excision repair system, the UV-induced DNA damage can accumulate, and that can cause mutations in the major or key genes that regulate cell division and apoptosis. So overall, as a whole, like DNA repair mechanisms, all three of these, they're very important for protecting the genome from damage and preventing the accumulation of mutations that could lead to serious health issues, including cancer and neurodegenerative diseases. And each type is very specific in its own way, treating specific types of DNA damage. And so when they are defective, the consequences can be severe, leading to like Lynch syndrome or xeroderma pigmentosum. And so understanding the mechanisms behind each of these, like not only sheds light on like how you can understand the disease, but also like potential targets for how you can fix it, or how you can actually target therapeutic interventions to improve patients' quality of life. So here is actually going into basically a diagram form of what I just said, like now the blue molecules. I know there's a lot of acronyms on it, but I really don't even look at those anymore. I just look at images, and I kind of understand. It's very important to understand the big picture. So the blue is the cancer biomarkers, and basically this diagram is providing you a comprehensive view or overview of the key DNA mechanisms that cells use to maintain their genomic integrities, right? And so it's basically telling you how the different types of DNA damage are recognized and then repaired by specific pathways. So let's look at the basic excision repair on the left. So start all the way from the left. So BER. Remember, this type of damage, this repair targets the damage of any single base damage or any single stranded breaks, and it would repair the small or and null and like the ones that are not helix distorting base lesions, like caused by oxidative stress, alkylation, deamination. And so there are a lot of enzymes involved. So for example, OGG1 is actually a glycosylase enzyme. That's what actually recognizes and removes the damaged base, and that creates the AB6 site. Basically, an AB6 site is a site where the base is missing. And in PARP, PARP1 and PARP1 and PARP2, those are proteins that detect the single-stranded breaks, and then they help recruit the other repair proteins. And then there's XRCC1, that's basically a protein that acts as a scaffold to coordinate the repair process. There's also DNA polymerase beta. That actually is what fills in the missing nucleotides, and FEN1 cleaves the flap that forms during the repair if there is any flap or any like basically polishing anything that's needed. And then there's ligase 3, and ligase 3 essentially seals the nick in the DNA backbone, and that completes the repair overall. It's very crucial, like it's very important. I would say BR is very important for repairing damage that could otherwise lead to mutations if they were left unrepaired, right? For nucleotide excision repairs, again, they treat bulky lesions and crosslinks that are caused by thymine dimers, usually caused by UV radiation or any other chemical agents. A similar process here. I won't get into too much detail for purposes of time, but again, the process is pretty similar. There's like I guess you could say there's like the enzymes that recognize, and there's proteins like the XP proteins. Those are the proteins that recognize the lesion and then they initiate the repair process. And there's endonucleases like ERCC1 and XPF, and they're the endonucleases that actually make the incisions, like snip, like little scissors on either side of the lesion. Those are like molecular scissors. And then there's like a sliding clamp, sorry, called PCNA, and that helps recruit the DNA polymerase. And then there's like polymerase, the DNA polymerase a delta or epsilon. Again, it's just a type of DNA polymerase, and that's what synthesizes the new DNA strand to fill in the gap. And then again, ligase 1 that seals the newly synthesized DNA into the existing strand. Again, that molecular glue. So if there is a defect, like we mentioned before, it could lead to xeroderma pigmentosa, and that also increases more sensitivity to UV radiation and increases your risk for skin cancer. Now for MMR, for mismatch mediated repair, pretty similar. It basically addresses any damages with mismatch, like base mismatches or any small insertion or deletion loops that occur during DNA replication, because of course it's not 100% perfect all the time. And this is relevant for Lynch syndrome, and if it doesn't work. And there's also something called homologous recombination and non-homologous end joining, and I haven't talked about that. Basically, these address double-stranded breaks. So the first three I talked about, the base excision repair, the MMR, the nucleotide excision repair, those three address single-stranded breaks. But if both strands on the DNA are broken, that's when you need to use homologous recombination. It's basically a very highly like accurate repair process that uses a homologous sequence as a template for repair, as typically from the sister chromatid during that replication process. And any defects in homologous recombination can actually lead to a predisposition in breast and ovarian cancer, particularly in people with the BRCA1 or 2 mutations. You might have already heard of that if you studied a little bit of breast cancer. And then there's NHEJ, which stands for non-homologous end joining, which also addresses double-stranded breaks. And this is basically a repair enzyme that directly ligates the broken DNA ends together without the need for a homologous template. It's more error prone than homologous recombination as a result, because you're just sort of filling in the gap and hoping for the best without actually looking at without actually looking at like the chromatid copy. NHEJ is used much more because it requires less effort, but there can be more errors as well, right? So you are becoming more efficient, but you are also sacrificing quality. And so errors in this pathway can lead to like more genomic instability and contribute to cancer development if it's not used sparingly. So this diagram overall, it's talking about the specific pathways that cells use to repair the different types of DNA damage that can occur during replication. And each one of them has very specialized proteins and enzymes that recognize and correct each of these lesions. And so it's very important to understand these mechanisms, again, because big picture, when you're zooming out, they are directly linked to various genetic disorders and cancer. And so the integrity of the genome relies heavily on these guardrails or on these repair mechanisms, because they help prevent the accumulation of mutations that could lead to disease. Because of course we know that there is a redundant and degenerate genome, but and genetic code. However, you do want to try to be at least like 95% right? Because you want to make sure that any mutations in your DNA are addressed for. So these are more guardrails to address it beyond just the degenerate DNA. So again, if mutations do occur and they are passed on, um, there are DNA mutations and how they can actually affect protein function as a whole. So here we'll actually explore the different types of mutations and how they alter the genetic code and their potential impact on the proteins that come from this code. So again, if all those guardrails fail, this is what happens. This is what leads to mutations that get passed on. So I'll also use like clinical examples like cystic fibrosis and also sickle cell anemia to talk about it to make it more interactive. But basically, mutations, like what is a mutation? A mutation is basically a change in the DNA sequence that can occur naturally or due to external factors like radiation or chemicals. And these changes can be as small as a single nucleotide or as large as a section of a chromosome. And the impact of mutations on protein function really varies depending on the type and location of the mutation within the gene. So I'm going to talk about three different types of genetic mutations. There's point mutations, deletions, and insertions. And each of these mutations actually alters the DNA sequence in different ways, leading to varying impacts on the resulting protein and potentially contributing to genetic disorders. So understanding these mutations is very important because at the molecular level, because it translates into disease to our patients. So let's start off with point mutations. A point mutation is basically a change in a single nucleotide base in the DNA sequence and is actually one of the simplest forms of genetic alteration, but it can have significant consequences depending on the location within a gene. So there are actually three different types of point mutations that I'll go into depth in the next slide, but there's a silent mutation, a missense mutation, and a nonsense mutation. And then there's deletions. A deletion mutation basically involves the removal of one or more nucleotides from the DNA sequence. And that can range from a single base to large sections of a chromosome. And there can be like a single nucleotide deletion or a larger deletion. And the single nucleotide deletion can cause like a frame shift if it occurs within the coding region. And that shifts the entire reading frame of a gene, leading to like a completely altered sequence of amino acids downstream from the mutation site. And an example of this type of frame shift mutation actually results from cystic fibrosis. And cystic fibrosis often results from a deletion of three nucleotides, leading to the loss of phenylalanine at position 508 in the CFTR protein. And this deletion causes the protein to misfold and malfunction. Remember, the misfolding happens because there's supposed to be like a positive, but it is supposed to be and instead there's like a negative. And remember, that's why knowing chemistry is so important because that's how you know when you understand hydrophilic and hydrophobic interactions within the cell, you understand why proteins misfold as well. And that actually leads to the symptoms of the disease of cystic fibrosis. And then if there's like a larger deletion that remove like entire sections of a gene, that can actually lead to a loss of function. So um, it could actually lead to a protein being non-functional, and that can have downstream effects as well. Now, the third type of mutation in the DNA is called an insertion, and the insertion mutation just adds one or more nucleotides. So basically, like the opposite of deletion, instead of deleting, you're like inserting one or more nucleotides. Um, and the impact of these insertions are similar to deletions, depending on the size and location within the gene. So for like one example I think of right now, like for a type of insertion, like a single nucleotide insertion is like Tay-Sachs disease. That's often caused by an insertion mutation that actually disrupts the reading frame. Because remember, the reading frame, I'll show you a video, the reading frame codons are read in sets of three nucleotides. Three nucleotides usually make up one amino acid. Again, see here on the chart, GAU that makes aspartate, or like GGU means glycine, but same with everything else on this chart, and in this box of the chart for glycine. But if you delete one of them, that can turn this into GUA, for example. GUA is a completely different protein that can lead to protein misfolding and eventually like a different disease. So one example of like a single nucleotide insertion that occurs that we learned is in Tay-Sachs disease. That's like caused by a mutation that disrupts the reading frame of the hexa gene. And that leads to the production of a non-functional enzyme and the accumulation of like harmful substances in the brain. And if there's a larger insertion, that can actually disrupt the normal structure and function of that protein. And it can have, of course, harmful functions. And remember, like point mutations, deletions, and insertions, they're all responsible for many different types of genetic disorders. It's not just Tay-Sachs, it's not just cystic fibrosis. And so the severity of the disease often correlates with the type, the location, and the size of the mutation. So understanding these mutations is very important for diagnosing and developing treatments for these conditions. And with advances in genetic technology, there are a lot of growing efforts to correct these mutations at the DNA level before you even like manifest the disease. That actually offers hope for any targeted therapies that can alleviate or even cure genetic diseases. So there is a lot of potential for therapeutic interventions now that we are studying it in more depth. So the main types of mutations, just to go into detail, like the silent mutations, again, this is the type of point mutation. Silent mutations often involve like a change in the DNA that does not alter the amino acid sequence of the protein. That actually happens because of, remember we talked about redundancy, where multiple codons can code for the same amino acid, and usually happens in the third part of the codon. And these silent mutations are silent. It's in the name. They have no effect on the protein function, but they can occasionally affect gene expression levels or splicing in very rare cases. And then there are missense mutations. And a missense mutation results again in the substitution of one amino acid for another one in the protein. And it doesn't affect the reading frame, but it affects like the folding, you can say. I'll give you an example to understand it a little bit better. But the effect of this mutation actually depends on the chemical properties of the substituted amino acid and its role in the protein. So a clinical example of this is sickle cell anemia, very classic example. Sickle cell anemia is actually caused by a missense mutation where the amino acid glutamic acid is replaced by valine in the beta globin chain of hemoglobin. And this single substitution actually leads to the production of hemoglobin S or HbS, which actually causes the red blood cells to become sickled in shape under low oxygen conditions. And these sickled cells are less flexible. They can actually block blood flow and also break down prematurely, and that leads to anemia, pain, and also other complications that manifest in symptoms for patients. Now, nonsense mutations actually introduce like a premature stop codon in the sequence, and that leads to the truncation. Remember, truncation means the stopping of the protein like prematurely. And that actually typically results in a non-functional protein. And basically the severity of this impact really depends on where the stop codon occurs in the gene. Earlier truncations of a protein tend to have more severe consequences because you didn't finish the protein all the way through, and depending on how much you didn't finish, that'll depend on the impact. But honestly, a lot of it, we do have mutations in our bodies, but a lot of them can be silent, or a lot of them can just, maybe I do have a missense mutation somewhere, but it's not in a gene that would lead to a disease manifestation. So that's why I would be healthy, just hypothetically speaking, because statistically speaking, we probably do have mutations, but overall, from all the guardrails that we talked about, the different DNA repair mechanisms, we are doing very well, indeed. So the last type of mutation in the DNA consequences section includes frame shift mutations that occur due to insertions or deletions of nucleotides that are not in multiples of 3. So this all, like, you can delete 1 or 2, and so that would actually completely shift the frame when you're reading this, and I'll show you what I mean by reading in a second, but because they're read in sets of 3. And an example of this is cystic fibrosis that we talked about before. Remember, the CFTR protein that's affected actually leads to like thick, sticky mucus in the lungs and the digestive system, and that causes the symptoms that are associated with cystic fibrosis. So, now this is actually taken directly from the first aid step book. You can see here in the top line says the degree of change. So you can see here that the least like effective or deadly is of course silent mutations, so they didn't really affect you at all. And then the most effective one or most defective one, or something that can most likely cause disease is frame shift mutation. And it talks about how like DNA polymerase and DNA ligase actually help are actually repaired the single point mutations. And actually, I wanted to also show you all the bottom graph here that kind of shows you a visual of how a silent mutation, even GAA or if it's like another thing that codes for GLU, that's still fine. If there's a missense mutation, it could actually lead to sickle cell anemia versus the diamine to uracil, or a nonsense mutation where there's a premature stop codon. Remember the UAG I talked about and all the other in that chart that I call it the codon dictionary chart. If there's a frameshift insertion that actually affects the multiple of 3, or frameshift deletion, which, if C is it's supposed to be the first letter of the second codon, instead it becomes the third letter of the first codon, and that affects the protein as well. All right. And again, I'm just giving you an overview. You can of course read this on your own time, and sort of it's a nice summary of what I just said. Now here is an overview of the functional organization of eukaryotic genes. Now, remember from my biology degree, I also had to learn a little bit about zoology and animals, but thankfully, in med school, you do not need to focus on that. The clinically relevant things are eukaryotic genes here. So the functional organization of eukaryotic genes emphasize basically how intricate our design is to allow for the precise regulation of gene expression. And understanding this organization is very important to appreciate how cells control their function and respond to various signals, and how disruption in the system can actually lead to diseases like cancer. So there's something called exons, and exons are the coding regions of a gene that are transcribed into mRNA and ultimately translated into protein. So the exons are actually part of the instruction manual's instructions that you can read and absorb. And basically the exons, they determine the amino acid sequence of a protein. And in a mature mRNA, once the mRNA is processed, I'll talk a little bit about that. The exons basically are what remain after the introns remain in the nucleus or are spliced out. So an intron is basically a non-coding sequence that's present in the gene, but they're removed from the pre-mRNA during the splicing process before the translation in the cytosol. So again, I'm still in a nucleus. So while introns don't encode protein sequences, they actually play very important regulatory roles, including influencing gene expression, mRNA stability, and the regulation of alternative splicing. And the mutations within introns can actually disrupt splicing, leading to improper protein formation. So if something is an intron, but it by accident looks like an exon and gets read, that's not right. There are also promoters, and promoters are DNA sequences that are located upstream of a gene, and they basically serve as the binding site, or basically the signal, yes, please, look at me, sort of site. They serve as a binding site for the RNA polymerase and the transcription factors, and it's very important for initiating transcription. And the promoter region essentially determines where the transcription begins, and it's crucial for the regulation of gene expression, because you don't want to be transcribing places that don't need to be transcribed, right? So it can actually be influenced by various different transcription factors that either enhance or suppress the expression of a gene. And if there are any mutations in the promoter region, it can actually lead to overexpression or underexpression of a gene that can potentially contribute to, again, cancer, you guessed it. And then there are enhancers. And enhancers, they are regulatory DNA sequences that can basically be located far from the gene that they regulate. They don't necessarily have to be right next to the gene that it wants to regulate. And they bind activator proteins that basically increase the likelihood that transcription of the gene will occur. And enhancers significantly increase the efficiency of transcription, and they allow for more fine-tuned control over gene expression in response to various signals in the cell. Because remember, our cells are not static and the environment is changing all the time. And they work in concert with the promoters to ensure that genes are expressed at the right levels, in the right cells, and at the right times, during development. So if there are any disruptions in enhancer function, it can actually lead to inappropriate gene activation or silencing, which actually contributes to diseases like cancer, like you guessed it. And so transcription factors are very important overall because they're essentially like proteins that bind to specific DNA sequences, such as the promoters or enhancers, to regulate the transcription of genes. And it's clinically relevant because of of the it's really very important to have tight regulation of gene expression. Um, because mutations in any of these regions, whether it's in the exons, introns, promoters, enhancers, or even the transcription factor binding sites, it can lead to misregulation of gene expression. And this misregulation is a common feature in many diseases, including, you guessed it, cancer. So again, understanding how eukaryotic genes are functionally organized actually helps you understand how genes are expressed and regulated. Now, let's talk a little bit more in depth about gene expression and its regulation. So, there's something called, so let's actually go in the order with transcription factors. So, we're going to basically focus on how gene expression is regulated at the transcriptional level, and there's something called an epigenetic modification. Now, epigenetic modifications play a critical role in normal physiology and also the development of various diseases. And so this regulation is this regulation basically ensures that genes are expressed in the right cells at the right time and in the right amounts. So let's actually break it down. So transcription factors, like we talked about before, have activators and repressors, and they fine-tune the regulation of gene expression, and very important for maintaining proper and appropriate cellular function. And then there's something called epigenetic modifications. And epigenetic modifications include DNA methylation. And DNA methylation involves the addition of a methyl group to the 5' carbon of cytosine residues. It's called, and it usually happens within something called CPG islands. And CPG islands are basically regions that are rich with cytosine and guanine nucleotides. And this modification typically represses gene expression by preventing transcription factors from accessing the DNA. So basically, methylation hides stuff. It basically hides parts of the gene that don't need to be activated. So remember what I talked about before? My skin cell has the genetic information to become a heart cell potentially, but the reason why my skin cell is not a heart cell right now is because the heart cell's genetic information is not expressed. It's methylated, it's silenced. So it has undergone an epigenetic modification so that what's in my skin stays skin. And it's very important to maintain that methylation for normal physiologic processes. But if there is abnormal DNA methylation, it can actually lead to inappropriate gene silencing or activation, which can contribute to cancer. So if there's, for example, like a hypermethylation of tumor suppressor genes, so if my tumor suppressor genes are turned off, that's like double negative, it can actually prevent the expression of tumor suppressor genes, and that leads to uncontrolled cell growth. There's also something called histone modification. And histones are basically proteins around which DNA is bound. So you can see here in blue, these little blue circles, these are called histones. And the DNA is bound around that histone. They form something called nucleosomes. And they're basically the basic units of chromatin. Remember, chromatin, chromosomes, DNA. And histone tails can undergo various like post-translational modifications. It's not limited. Like it can, you can have acetylation, methylation, phosphorylation, ubiquitination. Um, list goes on. So in gene expression, the histone acetylation is typically associated with gene activation. So if a histone is acetylated, it actually reduces the positive charge on the histones. Because remember, DNA is negatively charged. So for it to be tightly bound to a histone, it needs to be its opposite charge. So histones are positive. So does that make sense? Positive, negative attract. So histones are positive. But if a histone is acetylated, that will reduce the positive charge on histones, and that decreases their affinity for DNA, and it actually allows the chromatin to adopt a more open or transcriptionally more active conformation that actually, since it's exposed, it will most likely be transcribed, right? Whereas if it is tightly bound to a histone and it's methylated, or it's just super tightly bound, it's the DNA is going to be more silenced. So if there is a misregulation of histone modification, it is implicated in many diseases. So for example, like if there's too much histone deacetylation, it can actually lead to like silencing of genes that control cell cycle progression and apoptosis. And this it's actually one of the markers of cancer and metastasis. However, there's a lot of like epigenetic therapies that have been developed. So there are drugs that inhibit DNA methyltransferases or DNMTs and also histone deacetylases. They're both enzymes, remember they're ending in A's, and they're used to basically reactivate silenced tumor suppressor genes, which offer a very good therapeutic strategy for reversing the abnormal gene silencing. So like DNA methylation inhibitors, like there's a drug called decitabine, and that's basically used to inhibit DNA methylation in the treatment of certain cancers like myelodysplastic syndrome. And for HDAC inhibitors or HDAC inhibitors, there is a drug called romidepsin, and that's used to treat certain types of lymphoma by increasing the acetylation of histones. And that leads to the re-expression of silenced genes that can suppress the tumor growth, right? So again, a lot of clinical relevance to this molecular understanding of our DNA. So again, the epigenetic modifications, they add like another layer of regulation that allows it to have dynamic changes in gene expression in response to different environmental and developmental cues. But if these processes go go wrong, they can actually contribute to the onset of and progression of cancer and many other types of diseases. And so understanding these mechanisms is very important for developing like targeted therapies as well, because without an understanding, how are we going to fix it? So again, this is the DNA and histone modification sort of visualized. So there, again, there are three different types of modifications, which include methylation, acetylation, and phosphorylation, which highlights their roles in gene expression and also chromatin remodeling. And there, again, I talked about before, it's very important to regulate gene activity, and they both play significant roles in normal physiology and the development of diseases, such as like developmental disorders as well, besides cancer. So again, DNA methylation, we talked about CPG islands, how there's this involvement of the addition of a methyl group or CH3 to cytosine bases in the DNA at CPG islands that are rich in cytosine and guanine nucleotides. And that's usually catalyzed by enzymes that are called DNMTs or DNA methyl transferases. And that usually leads to gene silencing. So when DNA is methylated, it becomes tightly packed into something called heterochromatin. Heterochromatin is on in here. So on the left, your heterochromatin also tends to be much darker in molecular structure when you're looking at it in a electron microscope, whereas euchromatin is a little bit lighter. So that's sort of how you can also see the difference between them. But that is euchromatin and heterochromatin. We'll get into that later. But histone acetylation is also has to do with the involvement of an acetyl group, and acetyl is COCH3. And that acetyl group is usually added to lysine residues on the histone tails, which is carried out by histone acetyl transferases, and their acronym is fun. It's HATs, H-A-T, HAT, and then in plural it's HATs. Whereas histone deacetylases, the HDACs or HDACs, they actually remove acetyl groups. And acetylation actually neutralizes the positive charge on histones, like we talked about before, and that decreases the affinity for the negatively charged DNA. And that leads to a more open chromatin structure called euchromatin, like we see on the right here on this picture. And that euchromatin is much easier to be transcribed and considered genetically turned on, you could say. And that's associated with active transcription. And in chromatin remodeling, histone acetylation allows the transcription factors and other proteins access to the DNA, which facilitates the transcription of genes and is very important for development, differentiation, and also like respond to different environmental signals, like if you're sick or have a fever or something. And then there's phosphorylation. And so phosphorylation basically is usually happens to proteins themselves or enzymes. And you might have already learned about this in biochemistry, where phosphorylation involves the addition of a phosphate group or a PO4 to serine, threonine, or even tyrosine residues on proteins, and that's mediated by enzymes known as kinases. And dephosphorylation is the removal of a phosphate group, and that's carried out by phosphatases. And the phosphorylation of histones and transcription factors can either activate or repress gene expression depending on the context. And it's very important for signal transduction pathways where there's, positive feedback, negative feedback, and external signals that lead to the activation of transcription factors that regulate gene expression. And abnormal phosphorylation patterns are involved with cancer as well. And if there's like too much activation of kinases, that can also lead to uncontrolled cell growth and survival, as seen in many cancers. And kinase inhibitors are therefore a very significant class of cancer therapeutics that target specific pathways to control disease progression. So these DNA and histone modifications like methylation, acetylation, and phosphorylation, they are fundamental to the regulation of gene expression and the dynamic remodeling of the chromatin. And so there's this precise control, and that's very important for normal development and cellular function. While, like, if it becomes dysregulated, then it can lead to many diseases, and the pathophysiology of disease. So understanding these processes gives you like insights into how like the genes can be regulated, and then they can offer avenues for different therapeutic interventions in case the regulatory mechanisms don't work anymore. So remember we talked about the two main forms of chromatin and nucleus of eukaryotic cells. There's heterochromatin and euchromatin. So it's important to understand the differences between the two as well. So heterochromatin is much more highly condensed and tightly packed. And in the picture, you can see that the DNA is bound very closely around the histones that form a denser structure. And since the heterochromatin is generally like densely packed, it also means that it's transcriptionally inactive because it makes it difficult for transcription factors and RNA polymerase to access this DNA, which leads to gene silencing. And this form of chromatin is like often found in regions of the genome that aren't like usually transcribed, like the centromeres and the telomeres. And it plays a significant role, like heterochromatin plays a significant role in maintaining the genome stability by silencing like repetitive DNA sequences and transposons. And it's also very important to the structural integrity of chromosomes during cell division. And in contrast, the euchromatin is less condensed and more loosely packed. So in the picture here, you can see that the DNA is wrapped more loosely around the histones, and that allows for a more open and accessible structure. And euchromatin is associated with like active gene transcription, right? Because the open structure, it allows for the transcription factors and the RNA polymerase to easily access the DNA, and that leads to the transcription of genes. And euchromatin is typically found in the regions of the genome where the genes are like actively expressed, where it needs to like be transcribed ASAP. And it is important for like normal functioning of cells, and because it contains most of the genes that are transcribed to produce the proteins that are necessary for the various cellular processes. Um, and yeah, I think that's mainly it. I think what I can think of in terms of examples is like the formation of heterochromatin and gene silencing, because like heterochromatin, like in the X chromosome, basically females have two X chromosomes. So one of them is silenced to achieve dosage compensation, which involves the formation of heterochromatin. Um, and then for euchromatin, I can't think of one example at this time, but if I think of one, I'll discuss it. But again, the difference between the two is is stark and clear, and there is a contrast, and understanding the two because is important because both of them have implications in disease, but also normal physiology. Now here is our first complicated biochemistry cycle. We are going to go through all the complicated ones towards the end of the month in biochemistry when we talk about metabolism, but for now, this is the pathway of de novo purine and pyrimidine synthesis. Now remember, purines and pyrimidines are nucleotides. Nucleotides are the basic building blocks of DNA. So these pathways aren't just important for DNA replication repair, but they're also like significant targets in cancer treatment when things go wrong. So I'll break these pathways down for you and also discuss their clinical implications. So the key enzymes and steps in nucleotide synthesis, and I'll be very high level and I'll just talk about the more clinically relevant enzymes. So let's start off. Let's see here. Let's talk about let's talk, yeah, in yellow, the carbamoyl phosphate synthase 2, right? That forms from the top here. So this actually catalyzes the formation of carbamoyl phosphate from glutamine, carbon dioxide, and ATP. This is actually the rate limiting step in pyrimidine synthesis. And this pathway goes through many different like intermediates, including orotic acid to eventually form UMP. Your UMP stands for uridine monophosphatate, I believe. No, uridine monophosphate. And that can actually be further phosphorylated to form UDP and UTP. And UTP can then be made into CTP or it can be further converted to DTMP via the action of thiamidylate synthetase. Um, this enzyme, CPS2, is actually positively regulated by ATP and PRPP, and it's negatively regulated by UTP. And that provides this feedback loop or this feedback mechanism to control pyrimidine levels. And it's clinically relevant because there's a drug called 5-fluorouracil we talked about before, 5-FU, that actually targets this pathway by inhibiting thiamidylate synthetase, which prevents the conversion of DUMP to DTMP, and that's essential for DNA synthesis. So if you don't have DTMP, 5-FU basically like halts DNA replication, and so it makes it a very potent like chemotherapeutic agent, specifically in cancers like colorectal cancer. Now, in purine synthesis, the de novo synthesis of purine starts with PRPP or phosphoribosyl pyrophosphate synthase. Now, we can just use the acronym PRPP because it is a mouthful. Um, and the rate limiting enzyme for this pathway is GPAT. I'm going to try remembering what it stands for. I think it's like glutamine phosphoribosyl pyrophosphate aminotransferase, I believe. And that catalyzes the first committed step of purine biosynthesis. And this pathway leads to the formation of IMP after a bunch of different steps. This diagram doesn't even show them into IMP, which is a precursor for AMP or adenosine monophosphate, and GMP, which stands for guanosine monophosphate. And so GPAT is basically negatively regulated by the end products of AMP, GMP, and IMP. If there were too much of this, then there wouldn't be more GPAT being made here in this intermediate step. And there are a lot of drugs that target this pathway, including 6-mercaptopurine, I believe, 6-MP, and azathioprine. These drugs like inhibit purine synthesis by mimicking the purine nucleotides. And so that actually interferes with nucleotide metabolism and DNA replication because it kind of tricks the cell into thinking, oh, there's already enough of purines, I don't need to make more. And then methotrexate actually inhibits dihydrofolate reductase, which is an enzyme necessarily for the regeneration of THF or tetrahydrofolate. And that's actually a cofactor that's required in both purine and pyrimidine synthesis. So if you deplete the THF, the tetrahydrofolate, the methotrexate basically reduces the nucleotide synthesis, and that impairs DNA replication as well. And here you can see more drugs. So mycophenolate and ribavirin, they both inhibit IMP dehydrogenase, which is what converts IMP into GMP. That actually prevents the formation of GMP and therefore also reduces DNA and RNA synthesis. This is very important for DNA replication repair. Um, because without adequate nucleotide production, the cells can't replicate their DNA, and that can lead to cell cycle arrest and also apoptosis or programmed cell death. And it's very important in like rapidly dividing cells, like those found in the bone marrow, the gastrointestinal tract, and also tumors. So of course, if it is normal in the bone marrow and GI tract, you have this constant churning of new cells being needed to be made. So you would want to have this the de novo purine and pyrimidine pathways working there. But in tumors, they actually hijack this system and make it work for themselves, and that's when things go wrong. So you want to be able to target that and not the correct pathways, like in your bone marrow or your blood cells or your GI tract. But primarily, like 5-FU and methotrexate, they're both useful for chemotherapy for certain types of cancers. But one of the challenges of chemotherapy is really the development of resistance, where basically the cancer cells they adapt to survive despite the drug treatment. And this can actually occur through mutations in the target enzymes or the upregulation of alternative pathways or also the increased drug efflux. So this is why we don't have a cure for cancer yet. And hopefully you will be one of the people who contribute to a cure someday. Because these pathways are very important for normal cell function, especially in the rapidly dividing healthy cells like I talked about. So chemotherapy can actually have significant negative side effects to the normal functioning cells, including like bone marrow suppression. There can also be gastrointestinal toxicity and also hair loss for people who are taking chemotherapy. So now let's focus on something called the purine salvage pathway. And this is an important process that recycles purines from degraded nucleic acids and they form new nucleotides. So if there's any disruption in this pathway, it can actually lead to significant metabolic disorders, including ADA deficiency and Lesch-Nyhan syndrome that are highlighted here. So ADA stands for adenosine deaminase deficiency highlighted here, and understanding these pathways and the associated clinical consequences is important for diagnosing and treating these like genetic disorders. So very, very briefly, I would say the purine salvage pathway, it recycles like the free purine bases, right? The purines include the adenine, guanine, also hypoxanthine. They basically this pathway recycles it back into nucleotides, which can then be reused by the cell. And this is a much more energy efficient process compared to the de novo synthesis, especially in cells that have like high turnover rates, like lymphocytes or white blood cells. The key enzymes in these processes are HGPRT. HGPRT, I think, stands for hypoxanthine guanine something, phosphoribosyl transferase. But it will be most likely be called HGPRT on exams. And basically HGPRT converts hypoxanthine to inosine monophosphate or IMP, and it converts guanine to GMP or guanosine monophosphate. And the other key enzyme for this process is called APRT on the right hand in green, and that actually converts adenine to AMP or adenosine monophosphate. The key disorders, adenosine deaminase deficiency, ADA. ADA deaminase, remember it ends in an ACE, so it's an enzyme. ADA is basically an enzyme that converts the adenosine to inosine in the degradation pathway of the purines. And so if there's a deficiency in ADA, it can actually lead to an accumulation of adenosine, which is toxic to lymphocytes, and that leads to a severe form of immunodeficiency. And this condition, like ADA deficiency, is actually a major cause of SCID, or severe combined immunodeficiency. I don't know if you've heard of bubble boy disease, but that's basically what SCID is, bubble boy disease. And the people who are affected with SCID or bubble boy disease have a severely compromised immune system, and that makes them highly susceptible to infections. and how to treat it. Like you could treat it using like bone marrow transplantation, enzyme replacement therapy since it's ADA that's affected, and also gene therapy, which aims to correct the underlying gene defect. And Lesch-Nyhan syndrome is caused by the deficiency of the AGPRT enzyme. And this enzyme deficiency leads to an accumulation of PRPP and a decrease in GMP and IMP, right? Because they're being made less since there isn't the enzyme to convert the PRPP to the GMP or IMP. And that results in the increased de novo purine synthesis and the overproduction of uric acid. And this disease is actually clinical. The clinical features of Lesch-Nyhan syndrome include severe gout, kidney stones, also neurological symptoms like self-mutilation, also intellectual disability. It's very unfortunate because the overproduction of uric acid actually leads to hyperuricemia as well, and it's also associated with its own complications. Treatment for this actually focuses on managing the symptoms, particularly the hyperuricemia, with medications like allopurinol. Like a rheumatologist would be able to, prescribe these like that, or even an internal medicine doctor, like allopurinol, and that actually inhibits xanthine oxidase and reduces the uric acid production. But the neurological symptoms are difficult to manage, and they actually significantly impact the quality of life for the patient. And I think that those are the main points I wanted to hit for this slide. I just included a reminder of, purines and pyrimidines. Um, and um, and yeah, I think I hit on the all the main points. So basically understanding these like biochemical mechanisms and underlying conditions allows for again targeted therapeutic interventions and understanding like where exactly does like for example, allopurinol target, right? Allopurinol targets or inhibits the overproduction of uric acid eventually, right? So that's all I have. Like I really don't have this pathway memorized. I sort of just have the disease part memorized, right? Because that's what's clinically relevant for me in what's in green. Now let's talk about RNA to protein. So now that we've talked a lot about like nucleotides, DNA replication, a little bit about transcription, now we're actually going to get into the meat of the presentation. And now just as a review, this is from Khan Academy. Remember, transcription happens where? Transcription occurs in the nucleus and translation occurs in the cytoplasm. So remember, in transcription, you're going from DNA to mRNA, and in between there's pre-mRNA, and then it gets processed into mRNA, and then it leaves the cell's nucleus, and then it gets translated by a ribosome in the cytoplasm. And during translation, you convert the genetic information to proteins, and mRNA is actually code for the proteins that get synthesized at the ribosome. And remember, this codon dictionary I've shown it a couple of times, but this is actually how these triplet codons are. The tRNA sort of has the amino acid attached to it, and these are the arms. And it's eventually where the amino acids make up a protein from the growing protein chain. Now, I'll talk about this briefly for purposes of time, since we're running over time a little bit. But basically, pre-mRNA slicing has a spliceosome function. It has the formation of different complexes. And I think that's all I'll talk about for this. Basically, it's just good to have in mind that pre-mRNA does need to get spliced before it's ready to be an official, a big sister mRNA, a messenger RNA. Let me go to the next slide. Yes. And here's tRNA. tRNA stands for transfer RNA. This is very important to the translation process in protein synthesis. So understanding the structure and function of tRNA is very important to like understanding how the genetic information is actually translated into proteins. The tRNA structure has an anticodon arm, an acceptor stem, a T arm, a D arm, and a variable arm. And basically, the tRNA serves as an adapter that translates the genetic code into a specific sequence of amino acid. And each tRNA actually carries a specific amino acid that corresponds to the codon that it recognizes on the mRNA. So the tRNA with its attached amino acid or charged tRNA actually enters the ribosome, where its anticodon pairs with the complementary codon on the mRNA. And the ribosome then catalyzes the formation of the peptide bond between the amino acid on the tRNA and their growing polypeptide chain. The tRNA then basically like exits the ribosome and it leaves the newly formed peptide bond there, and the process continues until the entire protein is synthesized or when it reaches like a stop codon. Um, Sometimes there are mutations in the tRNA genes or in the sequences that regulate tRNA. Of course, that affects protein synthesis. So there are some examples, like some like neurodegenerative diseases have been linked to some abnormalities, and also some mitochondrial disorders have been linked to the mutations in mitochondrial tRNAs. These mutations can actually impair the mitochondrial protein synthesis, and that leads to defects in energy production and contributes to the pathology of some types of diseases. And then there's RNA polymerases. So basically, we're talking with all the key players right now, with the process of translation. We'll learn about translation. Basically, there's RNA polymerase 1, 2, and 3. They're each numbered in the same order that their products are used in protein synthesis. So from rRNA or ribosomal RNA, mRNA or messenger RNA, and then tRNA or transfer RNA. There's also RNA polymerase 4 and 5, but 4 and 5 are only in plants, not in animals. So again, here's where we're only learning clinically important information to the human body. So these three types of RNA polymerases are eukaryotic cells, which are responsible for synthesizing the different types of RNA, and they're critical for gene expression because they transcribe the genetic code from DNA to RNA, which is then used for protein synthesis. So RNA polymerase 1 is responsible for synthesizing ribosomal RNA. And a mnemonic to remember that, and ribosomal RNA is in the nucleolus. And the nucleolus is like that's the place where in the nucleus where ribosome production takes place. And RNA polymerase 1 is involved in making rRNA, which is the most rampant or most abundant RNA, which is a good mnemonic to use. And is used first in the process of protein synthesis that forms the structural framework of the ribosome, right? Because without the ribosome, you can't really translate anything. And then RNA polymerase 2 synthesizes mRNA, and also also makes microRNA or miRNA, and also small nuclear RNA or snRNA. The mRNA actually carries the genetic information from DNA to the ribosome, where it serves as the template for the protein synthesis. Because, of course, the DNA doesn't leave the nucleus. If it did, then the cell would not have the genetic information it needs to survive and function. That's why mRNAs exist. So while the snRNAs, they're mostly involved in splicing the pre-mRNA, and the microRNAs play a role in regulating gene expression. And RNA polymerase 2 also operates in the nucleus, where it transcribes genes into mRNA, which then undergo like processing before being exported into the cytoplasm. A mnemonic I use to try to remember what RNA polymerase 2 is because it's responsible for making mRNA, is that it's massive because of its central role in coding for proteins. And it's used second in the order of protein synthesis, and also produces regulatory RNAs like mRNA, like microRNA, and also snRNA. And finally, RNA polymerase 3 is responsible for synthesizing the transfer RNA and the 5S ribosomal RNA. tRNA, of course, we learned is important for translation because it delivers amino acids to the ribosome during protein synthesis. And the 5S rRNA is like the component of the larger ribosomal subunit because the ribosome has two subunits. And like the other polymerases, RNA polymerase 3 is also found in the nucleus. And I remember this through a mnemonic by basically saying like RNA polymerase 3 makes tRNA, and tRNA stands for like tiny, so like it's tiny compared to the mRNA and also the 5S rRNA. And it's also involved last in the events that lead up to protein synthesis, and because tRNA plays the role in translating, like physically translating the mRNA into a polypeptide chain. So first, there's rRNA that's produced by RNA polymerase 1 that forms the structure of the ribosome. Then there is the mRNA that is produced by RNA polymerase 2, and that carries the genetic code to the ribosome itself. And third, tRNA is produced by RNA polymerase 3, and that brings amino acids into the ribosome where they are assembled into proteins. Now here is sort of RNA processing in eukaryotes. There is a 5' capping. Basically, once the RNA is all made before before it's translated. There's a 5' cap that's added and also a poly A tail. It helps the mRNA become more mature. And without it, it can actually lead to the degradation of the transcript as the mRNA transfers from the nucleus to the cytoplasm. And here we're going to actually learn a little bit in depth of protein synthesis from initiation, elongation, and also termination.
Steps of the translation process. Initiation. The small ribosomal subunit binds to the initiator tRNA carrying the initiator amino acid methionine. This complex then attaches to the cap structure at the 5' end of an mRNA and scans for the start codon AUG. The process is mediated by several initiation factors. At the start codon, the large ribosomal subunit joins the complex and all initiation factors are released. The ribosome has 3 sites. The A site is the entry site for new tRNA charged with amino acid or aminoacyl tRNA. The P site is occupied by peptidyl tRNA, the tRNA that carries the growing polypeptide chain. The E site is the exit site for the tRNA after it is done delivering the amino acid. The initiator tRNA is positioned in the P site. Elongation. A new tRNA carrying an amino acid enters the A site of the ribosome. On the ribosome, the anti-codon of the incoming tRNA is matched against the mRNA codon positioned in the A site. During this proofreading, tRNA with incorrect anticodons are rejected and replaced by new tRNA that are again checked. When the right aminoacyl tRNA enters the A site, a peptide bond is made between the 2 now adjacent amino acids. As the peptide bond is formed, the tRNA in the P site releases the amino acids onto the tRNA in the A site and becomes empty. At the same time, the ribosome moves 1 triplet forward on the mRNA. As a result, the empty tRNA is now in the E site and the peptidyl tRNA is in the P site. The A site is now unoccupied and is ready to accept a new tRNA. The cycle is repeated for each codon on the mRNA. Termination. Termination happens when one of the 3 stop codons is positioned in the A site. No tRNA can fit in the A site at that point as there are no tRNA that match that sequence. Instead, these codons are recognized by a protein, a release factor. Binding of the release factor catalyzes the cleavage of the bond between the polypeptide and the tRNA. The polypeptide is released from the ribosome. The ribosome is disassociated into subunits and is ready for a new round of translation.
All right, I hope that sort of summarizes or maybe reviews for you the process of translation. And now we're actually almost done. Now we're going to talk about, once it is all translated, there's actually some post-translational modifications that occur, and they're basically these chemical changes that are made to proteins after they have been synthesized. And these modifications are important for regulating like protein function, stability, the localization of the protein, also how it interacts with other proteins. And it plays a key role in cellular signaling as well. So understanding these post-translational modifications is important for like comprehending like how these proteins actually execute their diverse roles in the cell and how the defects in these processes can actually lead to various diseases. So there's like phosphorylation, there's glycosylation, ubiquitination, and these different types of protein like post-translational modifications, they help for the functional diversity of proteins. And particularly like phosphorylation, it plays a central role in the cellular signaling pathways because if you're modifying key signaling proteins, the cells can then respond to external stimuli and like for example, like growth factors or stress signals that would enable like appropriate cellular responses. But on the other side, if there are like congenital disorders of like glycosylation, the degenetic disorders like they arise from defects in glycosylation, for example, and that would affect multiple organ systems. So post-translational modifications, they're vital for the proper functioning of proteins and the regulation of cellular activities. And these modifications allow the proteins to be fine-tuned in response to the cell's needs, ensuring that the cells carry out their roles effectively. But when these processes like go wrong, it can actually lead to significant clinical consequences. We've learned about cancer already, highlighting the importance of understanding post-translational modifications in both health and also disease. Now we're going to close out with watching a video sort of summarizing everything we talked about today from DNA to protein.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
All right, and that is basically how you go from DNA to protein, and then basically the foundation of how we operate in life. And from here, we don't have time to go through, I guess you could say, the cell specifically. I wanted to go through some like cell diseases, but hopefully next time we can sort of touch base on those and get some more clinical relevance out of them as to why we learned about every single cellular organelle and how it's relevant. So to be continued in our next part of our series of Biochemistry Month. But for now, we've opened the teleshadowing session quiz. So you'll need a 70% or higher to pass as always. So you can access that quiz at teleshadowing.com/certification. And next week, we'll be going over the diagnosing hereditary diseases. And one quick announcement is that we have also opened up all prior quizzes from our last 152 sessions So if you're interested in learning about a specific specialty led by a physician or wanted to sort of accumulate some more hours with our program, you are more than welcome to do so. It'll be under the shadowing sessions tab and also the certification tab. We have a spreadsheet, and we're also building some landing pages as well for every single specialty if you're interested in pursuing them someday. So thank you so much everyone for tuning in to today's session. We hope to see you at our next week's session. And